So today we will discuss DNA fingerprinting. The DNA fingerprinting, as we know, the fingerprint represents the individual identity, correct? No two twins also share the similar fingerprint. If জমজ হয় তাদেরও fingerprint সমান হয় না. But similar, like suppose a person has no hand, then who will collect the fingerprint? So there is a way that even though the person is absent, we can identify the person based on its DNA. Now each person has unique DNA pattern. This is called DNA fingerprinting. Using different methods, we have to identify, we have to distinguish the differences between two individuals in their DNA sequence. The DNA sequence of every individual has got subtle differences, very small differences, if not more. Say for example, between Tom Cruise and me, the difference might be very high, but Somodip and my difference will be DNA will be very less, but still there will be difference. Still there will be a difference. So how do you identify this difference? There are different methods for identification. One of those methods is RFLP. RFLP stands for Restriction Fragment Length Polymorphism. Restriction Fragment Length Polymorphism. It is also called in short as RFLP. REFLIP. RFLP in short. Okay? RFLP is short, it's called RFLP. Now to find out the changes in DNA sequence and to perform RFLP or RFLP, we must collect the DNA sample. We must collect the DNA sample. So extraction of DNA sample is very important. We can choose the source if the person is willing to give the sample, but if the person is unwilling, suppose in case of a homicide, correct? Homicide, the killer will willfully donate his DNA. You have to find out from the stray samples of biological samples around the victim. Now from that case, it becomes little bit more difficult. Otherwise, if we choose a soft tissue over a hard tissue, it is easier to extract DNA. Suppose we choose to extract DNA from bone, then it is very hard tissue. But if we choose the DNA from the squamous cell, it's very easy. From blood cells, it is very easy. So the buccal cells from inside cheek or mouth is taken for paternity test. Sources of DNA at crime scene may be blood, semen, hair follicle, saliva. DNA extracted from evidence is compared to DNA from known individuals. Correct? Suppose there has been a murder in a house, and in the house there are 4 residents. So you collect the DNA sample of 4 residents and then you collect the sample found in the nails of the victim. Correct? So if it matches with anyone, then the individual is or accused is murderer or killer. But is it 100% foolproof? Answer is no. There is a probability. There is billions of nucleic acid sequence. So if we go for permutation combination, you will find in millions and billions of time copies, one copy will get which will be completely identical. But getting that copy is highly impractical in normal scenario. Correct? Like if we all know that every 7 person in the world is Indian. But if you choose 7% from Heathrow Airport, there is what is the possibility? It's very less. And if you go to Kenya, if you choose any 7% from Kenya, it is very less to get that every 7% is Indian. Right? That's why. So this is theoretically possible, but practically it is impractical to find exactly identical DNA fingerprinting. Now, let me be very clear what is the difference between DNA fingerprinting and DNA footprinting. DNA fingerprinting is about sequences of nucleic acid or nucleotides, and DNA footprinting is a region on DNA which binds with certain proteins. It is a region where different protein binds to DNA, whereas DNA fingerprinting deals with only the nucleotide sequence. Correct? This is the difference. Now, as the DNA is extracted from the source, it is digested by known restriction enzymes. Restriction endonuclease hoche enzymes found in DNA in bacteria which cleaves a foreign DNA. Now it has specific site or restricted to specific sites where it acts. So if we take randomly any DNA sample from any organism, that restriction site may be present depending on the number of nucleotide sequence present in that particular site. Suppose it is a EcoR1 site. So it has got 6 nucleotides. So what the possibility is that we will get 10,000 times that particular sequence in human genome. Correct? So wherever this particular sequence is available, if we digest human genome with EcoR1, we will get those many fragments. Now, only one restriction endonuclease will produce less fragments, but it may be similar. If we take a combination of restriction endonucleases and then digest, then we get different segments of different lengths. Correct? Then we have to do electrophoresis. We have to arrange them according to their size from smallest to largest. Now, this term comes here. How? Suppose you take 2 individual species from this same two individual organism from the same species, like these 2 boys, if you take from human species, and if you digest, they are very closely related. They both are Bengalis. Suppose they both are from the same state, same district. They might be from the same parents. Now, if we digest their DNA with 2 or 3 different restriction endonucleases, we will see subtle differences. These differences is called reflip. That means, being human, in most of the cases the segments will be of same sizes with subtle changes, 1 or 2 nucleotide difference, 10 or 20 nucleotide difference in length, very small differences. More close they are in family tree, then less the difference, there is a possibility of having difference in their length of the polymorphism. Polymorphism means same DNA with different sizes. Okay? So we get different. This is called restriction fragment length polymorphism. So when 3 billion base pairs in the human genome, however, reflip analysis would produce a smear of many similar size fragments. Like I said, mostly it will have similar size fragments, except a few, very small region. How much small region? 1%. 99% will be same. Only 1% of the genome will have subtle differences in their length. Why this 1%? Why not 99% is different? Correct? This is how we were what we are now. So this 99% of our DNA contains, preserves, stores the proof of this root. So it takes 99% of genome identical to make us human being. Only 1% genome will slightly differ, where someone will become Elon Musk and someone will become you. Right? So we have difference in only 1%, because see, all human beings are not lactose intolerant. Right? We all are mammals, but we are not lactose intolerant, all human beings. Very few are lactose intolerant. So that within 1% only that will differ. Right? Within 1% only we will find thalassaemic patient. Within 1% only we will find diabetic patient. Within 1% only we will find type 1 diabetes, type 2 diabetes, coronary artery disease, stroke, everything. Early baldness, early graying of hair, correct? Weak bones, strong bones, muscular body, lean body, everything. Black hair, brown hair, golden hair, everything within that 1%. That 1% differs from Caucasians to African. We all are human beings, Asians to Hispanic group. Correct? So within that 1% only we will find the difference. 99% will remain same. And these particular 99% actually found is called junk DNA. So junk DNA is as important as having that 1% DNA, which makes sure that we have differences between two individual organisms. Because without the junk DNA, we will not be what we are now. If our junk DNA changes, then we would have become monkey. You got a you get a news in newspaper that so and so hospital one pregnant lady gave birth of one monkey, one snake, one crocodile. That 99% is responsible. Why? We are not crocodile or lizard. Okay? So that is why junk DNA is more important than that 1% DNA. Because there is a treatment. Suppose there are many treatments for diabetes, for graying of hair, for baldness, but is there a treatment that can cure, make you crocodile from crocodile to human being? No. So any defect in junk DNA, irreparable. Any defect in 1% DNA may be repaired. Now, the restriction fragment length polymorphism involves use of restriction enzymes. Now, these fragments produces a huge number of fragments from like a human genome like human genome, very long. So it is very difficult to find out very small differences. Correct? So we have other methods to identify these differences. One of them is VNTR, variable number of tandem repeats. Tandem repeats means a repeated sequence, a chosen sequence. Now we will try to find out in a given segment of DNA how many of those segments are present, those sequences are present. Let me explain clearly. In RFLP, we divide the DNA. This is the DNA. We divide the DNA like this. Another DNA. So the same group of enzymes produce 2 fragments in one DNA, so it's 3 fragments, and produce 4 fragments in other DNA. So 4, this is X, 3, this is Y. So X is different from that of the Y, correct? But problem arises when these fragments are now, suppose in both the individual the fragment lengths are same, then how? This method. So we take, suppose this is a sequence, suppose A, B, C, A. We will try to find out how many copies of these are present here and here, 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 here. Suppose the number is 2, 2, 0, 1, 1, 0. Now, is it different? Different? So now X can be differentiated from Y. This is variable number. The number, these tandem repeats are variable in number. In one of the segment is having 1, another segment is not having any of these tandem repeats. Right? This is how we distinguish two DNA molecules. Clear? So these segments are little bigger. The number of repeats at a given place on a certain chromosome is highly variable from one person to another. What does this particular sentence mean? When we are taking segments, then it is needless to scan the whole genome. We can choose a small segment of genome called chromosome. Take any chromosome, say chromosome number 15. Take the long arm of chromosome number 15, that is 15p. Short arm is 15q. So take a long arm of chromosome 15, and then find out how many copies of this particular tandem repeat is present. Give them, tag these tandem repeats with some radioactive elements so that they can go and bind and give radiation. You can visualize. Correct? So you will find in one chromosome of one person there are 5 places where it binds. It radiates. It shows radiation. In the other person it shows only 2 places. So this is 2 different. These 2 are different. Correct? This is how we find out difference between 2 individual DNA using VNTR, variable number of tandem repeats. The number of such repeats is usually different on the paternal and maternal members. Means we are born from our own father and mother. No one else infiltrated in between. How to prove? By this method, we can find out. You know? Suppose after your father's death, a boy comes to court of law and claims that he is also son of your father. and it came for the property. Then you have nothing to do but DNA test. If the DNA test you find that boy is not related to your mother, but related to your father, that means you have to share your property. Right? You have to share your property. Say this is the representation of VNTR. Just go through, just read it once and ask me any question you have. This is an important topic. You will get some other questions from this topic. Sometimes they will give all three methods and ask you to distinguish. Reflip, VNTR, STR, SINP. All four. Correct? Reflip, VNTR, STR, SINP. তাহলে প্রথমে আমরা শিখলাম reflip. 2 নম্বর কি শিখলাম? Number 3, STR means short tandem repeats. Short tandem repeats. What is this? Suppose there is a chromosome like this. We take this small portion of chromosome into consideration. Like VNTR is longer region. Now it may happen so that two DNA molecules may show same VNTR fragments or numbers, not fragments, numbers. It may happen so. It is just a probability. Then what we will do? We will reduce the size of the tandem repeat, make it shorter, hence the name short tandem repeats. So now suppose out of 10 nucleotides, what is the possibility of having 6 nucleotides in one place? Only one. But if we reduce the number of nucleotides to 2, then possibility to find out those 2 in 10 is at least 5. 5 times it can be same. So short tandems are the shorter sequences which we look for within a DNA segment. So now the short tandem repeat may show like this. Right? So one will show 4 segments, another will show only 3 segments. So now they can be differentiated. Right? Sometimes this also becomes identical. It may happen so. It's a question of permutation combination. It may happen so that they are so closely related. Suppose a twin sister are accused of murdering their mother or father or grandparents, anyone. Who has done it? How do you know? We do not have Eken Babu to solve the case. We have DNA. We do not have Feluda also. Who will solve? So DNA will solve. Now for closely related twins, homozygote twins, it is very difficult. They might have even STR similar, except single nucleotide polymorphism, SNP or SNP, single nucleotide. They differ only by single nucleotide. Now, this is very, very difficult to find out. This is a very cumbersome job. First of all, it is very difficult to find out the same region of the DNA of two individuals. After you find out and extract those regions, then you have to do capillary electrophoresis. This particular only one thing, capillary electrophoresis will help you. The capillary electrophoresis involves a capillary and it is at least 1 meter or 2 meter long, through which it is electrophoresed, where the DNA fragments differ, are separated even on the basis of single nucleotide. That means capillary electrophoresis provides higher and highest resolution compared to other electrophoretic methods. Capillary electrophoresis provides highest resolution. They can differ even single nucleotide. Right? So these are 4 methods by which we can distinguish or identify a particular DNA. Clear? Any question? So these are all different methods of finding a DNA. So DNA fingerprinting means you have to mention all 4, give their difference, give their use. What is the use? Disputed paternity test, identification of criminal, Number 3, diagnosis of rare genetic disorders. Diagnosis of rare genetic disorders. এই 3টে major use. Okay. Follow? Now I will stop. Any question you ask me. কতগুলো ছবি ছিল ওগুলো হারিয়ে ফেলেছি আমি। ওই ল্যাডার সিকোয়েন্স, ফাদার মাদার আর চাইল্ডের ল্যাডার সিকোয়েন্স কিরকম মেলে RFLP তে, ওই ছবিটা এখানে কোনো কারণে উঠে গেছে আর কি, খেয়াল করিনি। বলো, কোনো কোশ্চেন থাকলে জিজ্ঞেস করো। কোনো প্রশ্ন নেই? ঠিক আছে। তাহলে from Wednesday onwards, try to be present, full class, okay? It is good for you. You will enjoy it. You will see your friends delivering the seminar. Don't pull their legs. Rather, give them key. Boast them. Yeah? Cheer for them. Not by clapping or shouting. Okay. Slight clapping is okay. Not by shouting. Like in IPL. Don't make it an IPL ground, make it classroom. You'll enjoy it. You'll find, see, it is very easy when you see us delivering a speech, a lecture, a seminar from this desk, from this dais. But if I ask any one of you and ask you to deliver, I doubt how many of you will be able to even utter their name. When you see so many unknown faces, you will become conscious about first thing. Suppose you all start looking at this region, I will definitely have doubt whether my zip is open or not. Correct? So this is called manipulation by a crowd. Manipulation by a crowd. You will find this when you go to elite institutions. Elite institutions where most of the students are very meritorious. They behave very intelligently. It's very difficult to keep them, make them fool. It is very difficult to make them fool. We cannot deliver anything and everything just coming here on the dais. No. They won't allow. They won't allow. If you do not, if you are unable to deliver something which they get benefit from, they will make you understand. Even I am talking about even in undergraduate, not in postgraduate. But here, most of you do not even turn the pages of the book before coming to the class. That is why it is very easy for us to teach you. We can speak anything, cock and bull story. So, you can make a good teacher if you are good. The way you are, you will get the same society back to you. If you are corrupt, you will get corrupt politician, corrupt policeman, corrupt engineer, corrupt doctors. That's all. Can up to you, how you want to see your society 10 years from now. Things are changing rapidly. With the advent of AI, the job market is shrinking. Job market is shrinking. Wherever you have repetitive work that has been replaced by AI, will be replaced completely by AI. Even you have to learn AI. You have to learn. You have to. Okay, so just try to be present, all of you, and then কি বলে সবাইকে উৎসাহ দেবে তোমার বন্ধু বান্ধবকে যারা দেবে be positive, always say positive about it. Don't think about what you are speaking. You might speak wrong, you might speak less, more, you might fumble, you might forget. Don't worry, don't get panic. This is a thing you should be come out of. Now we are trying to push you through this so that in future you do not fumble in front of a crowd. Because as an Indian medical graduate, doctor, you will be asked to deliver a speech on thalassemia, on something, on diabetes, on snake bite, anywhere. You should be able to speak. Speaking means expressing. What you know, you must share. So how you share? How you speak? Remember, language proficiency is definitely matters. Since our education is mostly English based, so those who are good in English, they can deliver in better way. But that does not mean যারা বাংলায় ভালো বা ইংলিশে অতটা ভালো নয় তারা ইংলিশে বলতে পারবে না। Now, suppose in your class, how many of you are from Bengali medium or non-English medium? See? Bengali medium or Hindi medium. Only 3 or 4, correct? In our time, the scenario was just opposite. Out of 100 students, 80 were from Bengali medium. Only 10 or 20 will be from English medium. Correct? Now we are speaking English because we have learnt it. How? We read the book again and again. So read the book. Grow the habit of reading books. More you grow this habit, it is very, very helpful for you. If you do not read, you do not have the patience to go on reading. Only reading again and again, same thing. Science, medical medicine is nothing but perseverance. Perseverance. It is not about merit. It is not about IQ level. It is about how long you can maintain your patience. তোর সঙ্গে কথা বলবো, বাইরে বেরো। তুই নীল জামা, হ্যাঁ আয় না, তুই বেরো, তো দেখাচ্ছি তোকে। তুই আয়, ক্লাসে পড়ে আয় না, দেখা হবে তো তোর। কি নাম তোর? কি নাম? যাই নাম হোক, তোর নাম আমার জানা দরকার নেই, মুখটা চেয়ার রইল। তুই নাম নিয়ে বসে থাক, ছ্যাবলা আমি দূর করে দেবো। তুই তো খুব স্মার্ট। কত বললাম তোর? 101 দেখে নেব কি সেমিনার দিস। সেদিনকে অ্যাবসেন্ট থাকবে, দেখে নেব কি সেমিনার দিস। শ্যাবলামি পেয়েছো নাকি? ক্লাসের টিচার কথা বলছি, একটা জিনিস বলছি, একটি ভালো জিনিস। এটাকে ডিগ্রি কলেজ? বেরিয়ে ঝান্ডা নিয়ে বেরোবি? এটা একটা পার্ট এনএমসি কারিকুলামের। অর্ধেক ছেলে এখানে যদি দাঁড়িয়ে এসে বলতে বলা হয় না কিছু 10 মিনিট, প্যান্ট ভিজে যাবে। বসো। এগুলি করার আগে বড় হয়েছো, এগুলি করার আগে ভাববে দুবার। এটা কলেজে এসছো, প্রেস্টিজিয়াস জায়গা, সেখানে বসে খিল্লি মারছো। ওখানে এটা কি রক? চায়ের দোকান পাড়ার? মানে নিজের স্থান সম্বন্ধে কোনো গ্র্যাটিটিউড নেই, নিজের জায়গা সম্বন্ধে কোনো রেসপেক্ট নেই। আমার না রেসপেক্ট নাই বা করলি, আমার বয়ে গেছে তোর রেসপেক্ট নিতে। কলেজটা খারাপ করছিস তো। ছ্যাবলামিগুলো বন্ধ কর, বড় হয়েছিস তো। ছ্যাবলামি করতে হলে এখানে ভর্তি হলি কেন? পলিটেকনিক করতে হতিস, ছ্যাবলামি করতিস সারাদিন, কেউ কিছু বলতো না। কোন ডাক্তারকে দেখেছিস ছ্যাবলামি করতে রাস্তা দাঁড়িয়ে? যাও, ফালতু মুড অফ করে দেবে। So try to focus on, first you prepare the material, write up, okay? Then you prepare the PowerPoint. You can even present on chalk and board, doesn't matter. But how you do this? There are certain methods of expressing in PowerPoint. How? You cannot go on writing, describing the whole thing in the PowerPoint. You must mention only the important points, and in between the lines you must explain verbally. Like, say for example, I follow the same thing. There are people who are read, yes, I have seen taking the class. That is not the way. You must come prepared. Whether it's a class of 15 minutes, 30 minutes, or 1 hour, doesn't matter. You should be aware of this topic, what you are going to talk about. Give your everything to learn that particular topic. You will learn that even a small topic, which looks like a small topic, it has got immense depth. Whatever you ask, whatever you present, we will ask some question which you won't be able to answer. But it is not very difficult. It is there within what you have studied. It is not your failure. Don't see this as your failure. You failed to answer my question. No. This is a process of training that you should be able to face. you might not know the answer. Doesn't matter. You ask me anything, I might not answer, but I will say yes. Okay, I will check it and tell you later. That is the answer. That's all. That's all. Don't get panicked. Oh, if you ask her what will happen, nothing will happen. It's a training. So, purposefully participate in this training for your betterment. For your betterment, nothing else.